Hello, Triden and Nagar. Today we're going to be solving May-June 2023 Paper 1 Variant 1. And before we start, I'd highly appreciate it if you would take a moment out of your very busy day to subscribe to this channel and like this video. Now, attention all Cambridge AS biology students, we have some incredible news to share with you today. We are thrilled to announce the launch of our brand new website. And guess what? It comes with an exciting bonus, free notes for topics 1, 2, 3, and 4. Simply log in, you must log in. Then press file share to receive your free notes. Moreover, we have the AS Biology Help Group, which you are able to post any questions you may have. The link would be in the description box down below. Question number 1. The electron micrograph shows onion root cells prepared using a freeze fracture technique. The cells were quickly frozen and then physically broken apart. Freeze fracture breaks apart cells along weak areas, such as membranes and the surfaces of organelles. Which statements best explains the appearance of the electron micrographs? So let's see the suggestions that we have here. We have the endoplasmic reticulum, nuclear envelope, and the tonoplast. First of all, we must start by defining our terms. What is the definition of endoplasmic reticulum? It's a network of flattened sacs running through the cytoplasm of eukaryotic cells. Therefore, structure X that we're looking at is not the rough endoplasmic reticulum. Why? Because it's not a network of flattened stacks. A network of flattened stacks would look something like this. Exactly what we are seeing here. Therefore, structure X cannot be the endoplasmic reticulum. Now, let's see the rest of the suggestions. We must first zoom in deeper. What could you see here? Here we could see two layers. There's an upper layer and a lower layer, indicating that this is a bilayer. Therefore, this is a membrane which could be broken apart. Now, the second point, here we could clearly see pores. And as we all know, nuclear envelope is a bilayer with pores. Therefore, it makes sense that this structure X being the pores of the nuclear envelope. Therefore, the answer is going to be B. Now, the reason why this is not a tonoplast, despite it is an onion root cell, is because tonoplast is what covers the vacuole. And here we could see that it's a bilayer with pores. However, a vacuole or the tonoplast is only a single layer. And plus, this structure cannot be the plasmodesmata. The reason for this is because the vacuole, let's draw here a plant cell, the vacuole is covered with a layer of tonoplast. And the plasmodesmata is a pore at the cell wall. Therefore, D cannot be a correct answer. Therefore, the answer is going to be B. Question number 2, which cell structures can form vesicles? First of all, let's start with cell surface membrane. So we know that at the cell surface membrane, there are two processes that takes place, exocytosis and endocytosis. During exocytosis, substances leave the cell via fusing with the cell surface membrane, forming a vesicle, and then it leaves. Same with endocytosis. What happens is external substances fuse with the cell surface membrane, and then it goes inside via some vesicles. Therefore, cell surface membrane is correct. Now, endoplasmic reticulum. So, in the endoplasmic reticulum, the modified proteins are broken off from the cisterna and packaged into RER vesicles and moved to the Golgi body for modification. So, we know that the endoplasmic reticulum, or most particularly the rough endoplasmic reticulum, forms polypeptides. Therefore, what happens is that when the ribosomes found on the rough endoplasmic reticulum forms polypeptides, what happens is they are packaged into cisterna and then RER vesicles. These vesicles break off and goes to the Golgi body, and the function of the Golgi body is to modify the proteins. Therefore, endoplasmic reticulum is correct. And now, Golgi body. So, as we all know, Golgi body modifies proteins. Therefore, what happens is it forms Golgi vesicles after the modifications, which carry their contents to other parts of the cell, often to the cell membrane for secretion. The Golgi apparatus chemically modifies the molecules it transports. Therefore, both The endoplasmic reticulum and Golgi body have vesicles, therefore the answer is going to be A. Question number 3. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. Which student correctly matched the numbered function with the appearance of these cell structures? So let's see the suggestions that we have here. For 1, synthesis of polypeptides. We all know that ribosomes on the rough endoplasmic reticulum synthesize polypeptides. Therefore, the answer is going to be rough endoplasmic reticulum. Now, regarding to synthesis of lipids, we all know that the endoplasmic reticulum, the smooth endoplasmic reticulum, synthesizes lipids such as cholesterol. Therefore, 2 is going to be the smooth endoplasmic reticulum. For 3, packaging of hydrolytic enzymes that will remain in the cell. So as we all know, the Golgi body modifies proteins, therefore allowing the formation of lysosomes, which has the hydrolytic enzymes in a container so they don't cause damage to these cell organelles. Therefore, the answer is going to be the Golgi body. Now, let's see the suggestions that we have here. For 1, it's either W or Z. Now, let's look at W, non-membrane bound spherical structures. We all know that the ribosomes are not membrane bound and they are spherical. Therefore, this is ribosomes. And the answer is going to be W. Now for Z, membrane-bound sacs arranged as a flattened stack. So a flattened stack is the Golgi body. Therefore, this is incorrect. Now, for 2, let's see the suggestions. Now, let's start with V, membranes which surround an enclosed inner cavity. So an enclosed inner cavity forms a lumen. And that lumen is the smooth endoplasmic reticulum, forms a lumen. Therefore, the answer is going to be V. Now let's look at Z. We just said it's the Golgi body, so it's incorrect because Golgi body are in flattened stacks. Now for W, non-membrane bound spherical structures are the ribosomes, which we said they are incorrect. And regarding D, let's look at V, membranes which surround an enclosed inner cavity. It's a lumen. And we just mentioned that this is correct since this is the smooth endoplasmic reticulum. Now, for 3, let's look at Z. Membrane-bound sacs arranged as flattened stacks. We just mentioned this is the Golgi body. Therefore, the answer is going to be A. Question number 4. Which cells contain a tonoplast? So, tonoplast is the membrane which covers a vacuole, of course, in a plant cell. Now, let's see the suggestions that we have here. So, as a general rule, most plant cells do have a vacuole. However, there are some exceptions. Now, let's see the suggestions that we have here. Root hair. Yes, root hair cells do contain a vacuole since it's a mature standard plant cell. Now, companion cells, we know that it's a highly active plant cell. The reason for this is that it pumps hydrogen ions to its cell wall to facilitate the transport of sucrose within the phloem. Therefore, it must have a vacuole. Now, sieve tube elements. Here, that's an exception. Even though it's an alive cell, it lacks a vacuole. The reason for this is that sieve tube elements are a type of sclerenchymal tissue. And sclerenchymal tissue actually are dead as they mature, and their vacuoles and nucleus actually dissolves at the cell wall. Therefore, sieve tube elements lack a vacuole. And by common sense, we know that sieve tube elements facilitate the movement of sucrose, are stations along the phloem and facilitate the movement of sucrose, and they have pores. Therefore, it makes sense that it lacks a vacuole to facilitate the movement of sucrose and provide least resistance. Therefore, sieve tube elements do not contain a tonoplast which surrounds a vacuole. Therefore, this is correct. So as we all know, the function of the endodermis is to regulate the movement of water, ions, and hormones into and out of the vascular system. Therefore, it makes sense to have a storage such as the vacuole. Therefore, the answer is going to be D. Number 5. Which organelles found in animal or plant cells are surrounded by double membranes? Now, let's see the suggestions that we have here. A. Chloroplasts, which is correct. It is surrounded by a double membrane. Mitochondria is also a double membrane structure. and vacuoles. So vacuoles is incorrect. The reason for this is that it's surrounded by a membrane called a tonoplast, which is a single layer or a single membrane. Therefore, A is incorrect. For B, chloroplast, mitochondria, and nuclei. All three are correct. The reason for this is that nuclei are surrounded by a nuclear membrane, which is a double membrane. Now, as we said, vacuoles is incorrect, therefore the answer is going to be question number 6. Some scientists think that mitochondria evolved from bacteria that entered the cytoplasm of a different cell and were able to survive there. Which structural features of mitochondria support this hypothesis? So here we're talking about the endosymbiotic theory, which states that our cells evolved from prokaryotic origins. Therefore, here we're looking for prokaryotic features. Now, let's see the suggestions that we have here. Folded internal membrane. So, according to prokaryotic features, they do not have any membrane-bound organelles. Therefore, this is totally incorrect. Now, circular DNA. We know that bacteria or prokaryotes always has a circular DNA, which is not found inside a nucleus. So, this is correct. 70S ribosomes. This is correct. 70S ribosomes are prokaryotic, whilst 80S ribosomes are eukaryotic. Therefore, the answer is going to be D. Question number 7. What is present in all viruses, all prokaryotes, and all eukaryotes? A. Ribose sugar. So, ribose is a pentose sugar found in the nucleotides of RNA only. And deoxyribose are also pentose sugars found in the nucleotides of DNA only. Now, both of these are incorrect. The reason for this is viruses could either be RNA viruses or DNA viruses. Therefore, this is not always correct, and both of these are incorrect. Now, cytosine. Cytosine is a pyrimidine base found in both RNA and DNA. Therefore, this is correct. Now, thymine. Thymine is replaced with uracil in RNA. Therefore, this is not always correct. Thymine is only found in DNA, and we just mentioned that viruses could be either RNA or DNA. Therefore, this is incorrect, and the answer is going to be C. Question number 8. The table shows some steps that can be made in carrying out Benedict's test. So, Benedict's test is used to test for the presence of reducing sugar in a solution, and a positive test would be a red color change. Now, which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution? So, a semi-quantitative test is used to partially estimate the concentration of reducing sugars in a solution. Now, let's see the suggestions that we have here. So, for us to test a semi-quantitative test, we need to standardize the volume of Benedict's solution. Therefore, these are correct. Now, the second choice here is boil with hydrochloric acid and then neutralize with alkali. This is totally incorrect. The reason for this is once we boil with hydrochloric acid, what we're doing is that we're breaking the glycosidic bonds. And breaking the glycosidic bonds, now we're going to test for non-reducing sugars, which is totally incorrect. We do not want to test for the non-reducing. Now, next step is standardize boiling time with Benedict's solution and compare the final color with numbered color standards. So, since it's a semi-quantitative test, the color could indicate the concentration. So, with the red being the highest concentration of reducing sugars and green and yellow being the lowest concentrations. Therefore, this is correct, and the answer is going to be C. Question number 9. The diagram shows 3 examples of different bonds. Which bonds hold the secondary structure of proteins together? So, the definition of secondary structure are the hydrogen bonds formed between the peptide bonds of proteins. Now, let's see the suggestions that we have here. So, for bond 1, here we could see a peptide bond. So, a peptide bond looks something like this. Here, the oxygen, which is a partially negative charge, binds or forms a hydrogen bond with the hydrogen ion, which is partially positive. Therefore, this is correct. Now, for bond 2. Bond 2 is a peptide bond. Now, peptide bond is a covalent bond. And peptide bonds form the primary structure of proteins. Therefore, this is incorrect. Be careful. Here, we're forming hydrogen bonds between different peptide bonds. You can clearly see these are different proteins. However, here is the same. Therefore, this is incorrect. Plus, this is a covalent bond between the carbon and the nitrogen, which is the primary structure. So, bond 2 is incorrect. Now, for bond 3. Bond 3 is a tertiary or quaternary structure. The reason for this is that for tertiary structure, there's something called the disulfide bonds, which are also covalent bonds and hard to break. These form between the sulfur-containing R groups. Therefore, the answer is going to be C. Question number 10. Insulin is a globular protein involved in cell signaling. It is transported in the blood plasma from the cells that synthesize it to its target cells. A molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptide chains. Which statements about insulin are correct? So let's see the suggestions that we have here. 1. An insulin molecule has a quaternary structure. This is correct. The reason for this is that here it's mentioning 2 polypeptide chains, and quaternary structure is the bond formed between 1 or more polypeptide chains. Therefore, 1 is totally correct. 2. Insulin polypeptides are held together by 6 disulfide bonds. So be careful of the wording here. This is incorrect. The reason for this is because here it says a molecule, so both chains, contain 6 sulfur-containing amino acids. Therefore, if you were to assume that each chain contains 3, therefore, it is going to be 3 bonds forming between the 6 containing amino acids, so 3 in the first polypeptide chain and 3 in the second. Therefore, be careful here, this is incorrect. 3. Amino acids with hydrophobic R groups would be found in the center of an insulin molecule. So, the definition of a globular protein, it's a soluble protein. The reason for this is because it has the hydrophilic R groups towards the outside. Therefore, it's able to form hydrogen bonds with water and it is soluble. Whilst the hydrophobic R groups of amino acids are found towards the inside. Therefore, this is correct and the answer is going to be C. Question number 11. Which statement correctly explains why blood plasma can be maintained at a stable temperature? Now, let's define a few terms here. First of all, here is a new term called specific heat capacity and latent heat of vaporization. We must first define these terms. So, specific heat capacity is the heat required to raise the temperature of the unit mass of a given substance by a given amount. So, it's the heat required to raise a temperature. Now, latent heat of vaporization is the quantity of heat in joules required to convert 1 kilogram of liquid to vapor. So, here is the evaporation. We're talking about evaporation, which is totally incorrect. The reason for this is we're asking why blood plasma can be maintained at a stable temperature, not evaporate. We're not here talking about evaporation. Therefore, here we're talking about only specific heat capacity. Now, let's see the suggestions. It's either a low or a high specific heat capacity. Now, high specific heat capacity is going to be the correct answer. The reason for this is because blood plasma is maintained at a stable temperature. This means that it is hard to change, which also means that it requires more energy or more heat in order to raise the temperature. If it requires more energy in order to change its temperature, then it must have a high specific heat capacity, not a low one. Therefore, the answer is going to be B. Question number 12. Which region on the graph shows the activation energy of an enzyme catalyzed reaction? So, an activation energy definition would be the energy required to start a reaction. Now, as we all know, that enzyme actually lowers that activation energy, allowing reactions to happen at even lower temperature. Therefore, it makes sense that the activation energy of an enzyme catalyzed reaction would be lower than without an enzyme. So let's see the suggestions that we have here. So the activation energy is always the part that pops up in this graph. Therefore, we just mentioned that the enzyme has a lower activation energy, therefore the answer must be B. Question number 13. The graph shows the effect of an increasing substrate concentration on the rate of an enzyme catalyzed reaction. Which description of changes to the reaction conditions could result in the line Q if all other conditions were kept the same? So line P represents the result when the enzyme is used at optimum pH and optimum temperature. And line Q represents the result when the reaction conditions are changed. Now we would like to know the change that caused a slight decrease in the rate, however, reached the same maximum rate at the end. Now, let's first define two main things. Here we have a competitive inhibitor and non-competitive inhibitor. Now, the definition of a competitive inhibitor is that it binds to the same place as the active site, therefore slowing the rate of the reaction. However, this is reversible. The reason for this is that if we keep increasing the substrate concentration, the increased substrate concentration is able to overcome the effect of the competitive inhibitor since it binds at the same site of the active site of the enzyme. Therefore, what happens is that the rate of the reaction slows down at first. However, because we increase the substrate concentration, it is able to overcome the effect of the competitive inhibitor and reach the same rate of the reaction at the end. Therefore, Q must be a competitive inhibitor added. Now, the definition of a non-competitive inhibitor is that it binds to an allosteric site. The definition of allosteric is a site other than the active site. Now, what happens in this case is that the enzyme shape is completely distorted once it happens. It binds to an allosteric site. Therefore, what happens is that the enzyme is denatured. This means it can no longer increase the rate of the reaction or facilitate any reactions. Therefore, what's going to happen is that the rate of the reaction is going to keep slowing down, and the rate of the reaction is going to be at plateau at a way lower rate than the maximum rate of reaction. Is going to be somewhere here. And the substrate concentration does not overcome the effect of the non-competitive inhibitor since it binds at an allosteric site, completely distorting the shape. Now, let's see the suggestions that we have here. For one, add an inhibitor that attaches to a site other than the active site. So here we're talking about a non-competitive inhibitor, and this is totally incorrect, and we've just mentioned why. 2, add an inhibitor that has similar shape to the substrate. Here we're talking about a competitive inhibitor which binds to the active site. Therefore, it has a similar shape to the substrate. Therefore, this is correct. 3, add an inhibitor that blocks the active site of the enzyme. Yes, this is correct since this is also a competitive inhibitor. 4, carry out the reaction at a higher temperature. So this is not absolute. The reason for this is because here line P represents the result when the enzyme is used at its optimum temperature and optimum pH. Now, optimum temperature is the best temperature with the highest rate of the reaction. What happens is if we operate at an even higher temperature than the optimum, what happens is that the rate actually decreases and lies plateau. The reason for this is that if we increase the temperature, the enzyme is actually denatured. Therefore, this is incorrect and the answer is going to be D. Question number 14. Which statement about cell signaling is correct? Now, A. One type of receptor molecule will recognize all ligands in the body. So, this is incorrect because a receptor molecule is specific for only one ligand, one shape. Therefore, this is incorrect. B. The binding of a ligand may cause a change to the shape of the receptor. This is correct because when a complementary ligand binds to its receptor, which is the same shape, what happens is that something called conformational change takes place. The receptor changes shape, and then the signaling cascade happens inside the cell once the receptor changes shape. Therefore, this is correct. For C, the receptors for ligands are always found inside of cells. This is incorrect. The reason for this is that there are extracellular receptors and intracellular receptors. For example, at the nuclear membrane, which are intracellular receptors. Therefore, this is not absolute and this is incorrect. D, the same ligand is made by all the cells in the body. This is totally incorrect because each cell has a completely different ligand and completely different products. Therefore, this is incorrect and the answer is going to be B. Question number 15. 4 students observed plant epidermal cells that had been placed in a concentrated sucrose solution for 30 minutes. They were asked to identify the partially permeable layer and to explain the appearance of these cells in terms of water potential and movement of water. Which student is correct? So, we must identify that partially permeable membranes are the cell surface membranes only, since cell walls are freely permeable. Therefore, these are incorrect. Now, water potential at the start and the movement of water during the experiment. Now, cell contents have a lower water potential than the sucrose solution. We will not know if that's the case until we look at the movement of water during the experiment. So, if the cell contents have a lower water potential, this means that they have a higher sucrose concentration. Therefore, this means that water will enter the cell down a water potential gradient. Therefore, this is incorrect. Now for B, cell contents have a higher water potential than the sucrose solution. Therefore, it has a lower sucrose solution and a higher water potential. Therefore, water would actually move out of the cell down a water potential gradient. Therefore, the answer is going to be B. Question number 16. The table compares the surface area to volume ratios of 5 agar blocks that differ in dimensions, but which all have the same volume. Which prediction can be made about the way in which size and dimensions of these blocks affect the efficiency of diffusion? So let's see the suggestions that we have here. A. The efficiency of diffusion will decrease as the width of a block increases. So let's see the suggestions that we have here. So regarding the width, here we could see that the surface area to volume ratio was the highest at the highest width. However, if we compare a smaller width, it's going to be 1.3. Now, if we go to 8, it's going to be 0.75. However, it increases again when we go to a lower width, which is 4 to 1.1. Therefore, this rule is incorrect. It is not absolute. Now, for B, the efficiency of diffusion will increase as the height of a block increases. Now, first of all, I forgot to identify a term. So, the efficiency of diffusion is the best when the surface area to volume ratio is the highest. Now, the efficiency of diffusion will increase as the height of the block increases. Now, let's see again. So, the greatest height is 8. Now, surface area to volume ratio is the lowest, which is not a good sign. Now, if we move on to 4, it increases again. And then 2, it increases, which is incorrect since the same heights have 1.3 and 1.5. So this is not absolute and this is incorrect. C. The efficiency of diffusion will increase as a block of fixed volume is flattened. Now, here what is it trying to say is that as the length increases and the width decreases or the height decreases, then the efficiency of diffusion will increase. AKA the surface area to volume ratio will increase because as this increases, the efficiency increases. Now let's see if this is correct. So for you to visualize it, we have a cube which is squashed, aka flattened. Therefore, we're looking for the agar blocks that has a lowest height and the greatest length, which is block 5. Since here the length is 64 and the width and the height is 4 and 2. And the surface area to volume ratio is 1.5. And same thing with block 4. We can see here that it has a greater surface area to volume ratio as the height decreased or it flattened. Therefore, this is correct. Now for D, the efficiency of diffusion will decrease as a block of fixed volume is elongated. So elongated is the same as flattened. However, it will not decrease, it would actually increase, since there is a greater surface area to volume ratio when it is flattened. Therefore, the answer is going to be C. 17. The cell cycle includes mitosis. What are features of this type of nuclear division? So, let's draw out the cell cycle to visualize it. So, the cell cycle includes mitosis. Now, mitosis is PMAT, part of the cell cycle, being prophase, metaphase, anaphase, and telophase. And this is the mitosis. After mitosis, there is a phase called cytokinesis, which the cells divide, forming two identical cells, which are smaller in size than the parent cells. Now, here, there's a phase after that called the interphase. Now, the interphase, what happens is there's the G1, S phase, and G2. In G1 phase, these cells which have just divided continue to actually grow and increase in size. Therefore, it comes to the S phase, which is the DNA replication phase. Replicates, and then in G2 phase, it does continue to grow. Now, let's see the suggestions that we have here. 1. Form cells of equal size to the parent cell. This is incorrect because in the cytokinesis phase, what happens is that both cells divide, or the parent cell divides into two identical cells. Now, the parent cell is going to be a large cell. It splits almost in the PMAT or in the telophase or cytokinesis into two smaller cells, which continue to grow in the G1 and G2 phase. Therefore, this is incorrect. 2. Forms genetically identical nuclei. This is correct. The reason for this is a single parent cell splits into two. Therefore, it makes sense to have one copy of each chromatid. Therefore, this is correct. 3. Semi-conservative replication of DNA. I just mentioned that this is incorrect since it's asking about the phases in mitosis. And the DNA replication takes place at the S phase, aka the interphase. Therefore, this is incorrect, and therefore the answer is going to be D. Question number 18. A student observed these cells in a growing region, meristem, of an onion root and obtained the data shown. Which percentage of cells contains chromosomes that appear as two chromatids? Now, let's see the suggestions that we have here. We have interphase, prophase, metaphase, anaphase, and telophase. Now, as we all know, the cell cycle consists of mitosis and interphase. The mitosis phase being the PMAS or prophase, metaphase, anaphase, and telophase. Then after that phase is the cytokinesis, which is the division of the parent cell into two. Then the G1 phase, which is part of the interphase, the S phase, where DNA replication takes place, and the G2 phase. And here is the cell cycle. Now, which percentage of cells contains chromosomes that appear as two chromatids? AKA the chromosome before the division or the attachment of the spindle to the kinetochore, separating the two chromatids to the opposite poles of the cell, aka in anaphase. Now, here we're going to have the prophase and metaphase, since it's before the separation by the attachment of the spindle to the kinetochore and then the movement of the chromosomes to opposite poles in anaphase. Now, so prophase and metaphase are correct. Now, the reason why interphase is incorrect, because in G1 phase, when the division of the parent cell takes place, each parent cell is going to have one chromatid only until the S phase. The S phase, once DNA replication takes place, now each chromosome has two chromatids only after the S phase. Therefore, here it didn't specify, it said only interphase. So this is incorrect. The reason for this again is because at G1 phase, there is only one chromatid up until the S phase. Therefore, this is inaccurate and incorrect. Therefore, what we're going to do is we're going to add the prophase to the metaphase. Therefore, 73 plus 16. And then we divide 89 by 1000 since it's the total of all number of cells, which is going to give us 8.9% after we multiply by 100. Therefore, the answer is going to be B. Question number 19. Which statement about messenger RNA is correct? A. In eukaryotic cells, mRNA is made by removing exons from the primary RNA transcript. So in eukaryotic cells, mRNA is made by removing introns. So the mature RNA is made by removing introns from the primary RNA transcript. Therefore, we're left with the exons at a process called splicing. Therefore, this is totally incorrect. B. mRNA is a single-stranded polynucleotide containing a different purine base than DNA. So, first of all, it contains a different pyrimidine base than DNA, and that pyrimidine base is uracil instead of thymine. Therefore, this is incorrect. See, mRNA molecules contain ribose sugars joined to phosphate groups by phosphodiester bonds. So, this is correct. mRNA contains ribose sugars, which are pentose sugars found only in the nucleotides of mRNA, contrasting DNA, which is formed of deoxyribose sugar. Be careful of this point. And yes, they are joined to phosphate groups by phosphodiester bonds between the phosphate part, since a single nucleotide is made by a nitrogenous base, a pentose sugar, which could be a deoxyribose or a ribose sugar, if we're talking about mRNA, and a phosphate. What happens in a phosphodiester bond is that phosphate, which is negatively charged, binds with the ribose, the OH part of the ribose sugar of another nucleotide. Therefore, forming a phosphodiester bonds. Therefore, this is correct. Now, for D, the monomers of mRNA contain a phosphate group, deoxyribose sugar, and a nitrogenous base. We just said that this is incorrect. Deoxyribose sugars are present only in DNA nucleotides. Therefore, this is incorrect, and the answer is going to be C. Question number 20. Which structures are involved in transcription only? So, transcription is the process of forming the mRNA molecule. Now, DNA template strand. This is correct. The DNA template strand is needed to form the mRNA molecule by the RNA polymerase aligning the mRNA or the RNA nucleotides together, forming the mature mRNA. Therefore, this is correct. Now, anticodons. Anticodons are incorrect since these are the nucleotides found on the tRNA, and tRNA is involved in translation, and they are found in the cytoplasm, which come to the ribosomes for the mRNA molecule to be translated. Therefore, they are incorrect. Now, RNA polymerase is correct since it is needed to align the RNA nucleotides along the DNA template strand to form the mature mRNA. Therefore, the answer is going to be B. Question number 21. One gene provides the code for the production of which type of molecule? First of all, let's define what a gene is. A gene is a small section of DNA on a chromosome that codes for a particular sequence of amino acids to make a specific protein, aka a sequence of many bases. We know that bases are made of 3 nucleotides. So one base equals 3 nucleotides. And we know that one base codes for an amino acid. And we just mentioned that genes are a collection of nucleotides or bases. Therefore, they code for many amino acids, aka polypeptides, or proteins. Therefore, the answer is going to be D. Question number 22. The table shows some mRNA codons that code for certain amino acids. What would be the change in the amino acid if the first base in the fifth DNA triplet was substituted for an adenine base? So, a DNA triplet is composed of 3 nucleotides, and a base is a part of a single nucleotide, since a nucleotide is made of a nitrogenous base, a pentose sugar, and a phosphate group. Now let's look for the fifth DNA triplet. So the first, second, third, fourth, and fifth. So here, GCA were substituted for ACA. Now the issue with this is that it's a DNA template strand, and the key here is within the RNA template strand. Therefore, we must convert the DNA to RNA via complementary base pairing. So, GCA was substituted to ACA. Now, converting this to RNA, it would be CGU, uracil, since RNA doesn't have thymine, and UGU. Now, let's look at the key here. So for CGU, we have arginine, so the substitution was from arginine to UGU, which is cysteine. Therefore, the answer is going to be C. Number 23. Which structures contain cytoplasm with mitochondria and a nucleus? So let's see the suggestions that we have here. We have xylem vessel elements. This is totally incorrect for both. The reason for this is that xylem vessel elements are hollow, dead cells. Therefore, they lack both a nucleus and a mitochondria. Therefore, this is totally incorrect. Now, companion cell. Companion cell do have a mitochondria, since it must generate ATP to pump hydrogen ions out to facilitate the movement of sucrose into the phloem sieve tube elements, so it does have a nucleus and a mitochondria. Now for phloem sieve tube elements. So phloem sieve tube elements actually lack these, since the companion cell actually carries out all the active processes. Therefore, this is incorrect, and the answer is going to be A. 24. What is the correct term to describe the intermolecular hydrogen bonding between water molecules? So, first of all, we have two forces carrying water molecules up the xylem. The first one is cohesion, the second one is adhesion. Cohesion is the hydrogen bonding between water molecules, so the answer is B, which is cohesion. Now, adhesion is the hydrogen bonding between the water molecules and the xylem vessel elements due to its hydrophilic properties. Therefore, the answer is going to be B, cohesion. Number 25, the diagram shows the outline of a xerophytic leaf that had been left for 45 minutes in different conditions, P and Q. Which statements about the cells in layer Y of the leaf in each of the conditions P and Q after 45 minutes are correct? First of all, since this is a xerophytic leaf, we will assume that the layer Y on top is the thick cuticle, since xerophytic leaves have a larger, thicker cuticle to prevent water loss. Therefore, Y is a thick cuticle. Now, let's see the suggestions that we have here. 1. There is a less negative water potential in P than in Q. So a less negative water potential means basically a higher water potential in P. This is correct since it appears turgid. Therefore, this is an indication that it has a higher water potential. For 2, the cells may be turgid in P and plasmolized in Q. This is correct, since Q is curved, which means that it has lost water and is plasmolized. Therefore, this is correct. 3, the cells are less turgid in P. This is incorrect, they are more turgid. 4, there is no net diffusion of water into Y in either P or Q. Yes, this is correct. The reason for this is I told you to assume, since we have a large structure at the surface, that this is a thick cuticle. And as we all know that thick cuticle are actually hydrophobic. They do not allow the diffusion of water, and their function are to prevent water loss from the leaf. Therefore, this is correct, and the answer is going to be A. Number 26. How does sucrose move from chloroplast to the phloem? So first of all, we must know the pathway of movement of sucrose into the sieve tube elements. So first of all, we know that we have a companion cell. The function of a companion cell is to pump hydrogen ions to its cell wall via hydrogen pumps. Then hydrogen ions combined with sucrose found outside, and they both go back into the companion cell via carrier proteins. And then once the sucrose is inside the companion cell, it diffuses via passive diffusion into the sieve tube elements. So now let's see the suggestions that we have here. 1. Diffusion. This is correct. We said that passive diffusion takes place once the sucrose is found inside the companion cells and then goes into the sieve tube elements, so that's correct. 2. Apoplast pathway. So the definition of apoplast pathway is the movement of water molecules via the cell walls of plants. And I've just mentioned that companion cells pumps hydrogen ions to its cell wall. Therefore, the apoplastic pathway does take place here, so this is correct. 3. Symplast pathway. So the symplast pathway contrasts the apoplastic pathway. The symplast pathway is the movement of water across the plant via pores called the plasmodesmata. Therefore, we just mentioned that companion cell and sieve tube elements are beside each other, and what happens is that the sucrose found inside the companion cell diffuses into the sieve tube elements. How does that happen? Via the symplastic pathway from pores called the plasmodesmata. Therefore, this is correct, and the answer is going to be A. Number 27. How are companion cells involved in loading sucrose into phloem sieve tube elements? So we just explained the process, however, we will explain it again. So, companion cells pumps hydrogen ions via proton pumps or hydrogen pumps to its cell wall. Therefore, what happens is that sucrose found outside combines with hydrogen ions and goes back into the companion cells via carrier proteins. Obviously, this is by facilitated diffusion. Now, once the sucrose is inside the companion cell, it diffuses by passive diffusion into phloem sieve tube elements via plasmodesmata. Now, let's see the suggestions that we have here. A. Actively through co-transporter proteins and passively through the plasmodesmata. Now, this is correct because the movement through the co-transporter proteins is active. The reason for this is that the companion cells pumps hydrogen ions via proton pumps, and then once the hydrogen ions are outside, they combine with sucrose, and then they move back into the companion cell. So for hydrogen, down concentration gradient, and sucrose against concentration gradient. Therefore, this is correct. And passively through the plasmodesmata. So we just said that once the sucrose is inside the companion cell, it moves by passive diffusion through the plasmodesmata or the symplastic pathway into the phloem sieve tube elements. So this is correct. B. Actively through co-transporter proteins and plasmodesmata. This is incorrect. It's passively through the plasmodesmata via simple diffusion. C. Passively through co-transporter. This is incorrect. And actively through plasmodesmata. This is incorrect. And the answer is going to be A. Number 28, the photomicrograph shows blood cells as seen using a high-power light microscope. Which row correctly identifies the different types of white blood cells? First of all, here we could identify a lymphocyte. The reason for this is that lymphocytes have a nucleus that fills the entire cell, almost the entire cell. Therefore, we can identify this one as a lymphocyte. Now, the second point is that lymphocytes are always smaller in size when compared to other white blood cells. Therefore, 3 is definitely going to be a lymphocyte. Now, regarding 1, it must be a neutrophil. The reason for this is that neutrophils are segmented, bilobed or trilobed nucleus. Therefore, this is a neutrophil. Now, regarding 2, This must be a monocyte or a macrophage since they are the largest type of white blood cells. Therefore, the answer is going to be C. Question number 29. Plan diagrams of 2 blood vessels are shown. Which labels are correct? So, first of all blood vessels have an inner layer called the endothelium. Tunica media and tunica externa. And the tunica intima contains the endothelium, which is the inner layer. So the inner layer is also the tunica intima. Now let's see the suggestions that we have here. So first of all, 1 and 2 and 3 are actually hard to identify. However, we could clearly see that 2 has a wider lumen, therefore indicating that 2 is a vein, versus 1 which could be an artery. However, we still don't have enough detail. Now, let's look at 5. I've just mentioned that the inner layer, or the tunica intima, is made of an endothelium. Therefore, this must be the correct answer. Why? Because epithelium is actually incorrect. This is found in plants. Therefore, this is totally incorrect. And squamous cells are found in the alveoli. Squamous epithelium. Therefore, this is totally incorrect. And epithelium, as we said, is found in plants. Therefore, the correct answer must be B. Question number 13. What are found in blood and tissue fluid? So, let's look at the suggestions that we have here. First of all, let's define what is tissue fluid. So, we have blood inside blood vessels. What leaks out? These blood vessels actually have pores. What leaks out is something called tissue fluid, which has less proteins and is less dense than blood. Now, let's see the suggestions that we have here. So, we have carbon dioxide. Yes, since tissue fluid leaks out, to provide the cells with the nutrients and the oxygen it needs, there are waste products from these cells, such as carbon dioxide. These waste products also dissolve in the tissue fluid to be transported back into the blood, so this is correct. 2. Fatty acids. This is also correct because fatty acids are part of the nutrients needed for cells to survive. Therefore, it does leak out in the tissue fluid and is both found in the blood and tissue fluid. 3. White blood cells. Yes, white blood cells are both found in the blood and they leak out in order to find or destroy infections and destroy pathogens. Therefore, this is also correct. And white blood cells are also small enough to be able to pass. Via a process called chemotaxis, they are able to move from the blood into the host cells. Now, for 4, proteins. Now, blood has a higher proportion of proteins and contains a protein called albumin. Which is too large to pass into the tissue fluid. However, there are other proteins found in blood which are small enough to be able to actually pass outside. The reason for this is also proteins are required for the normal growth of cells. Therefore, all of them exist and the answer is going to be A. Question number 31. Which reactions would be slowed down by an inhibitor of carbonic anhydrase? First of all, carbonic anhydrase is an enzyme found inside red blood cells. Its function is to combine carbon dioxide with water and convert it within a reversible reaction into carbonic acid. Therefore, what happens is that carbonic acid dissociates into hydrogen ions and bicarbonate ions. Now, let's see the suggestions that we have here. 1. Carbon dioxide plus hemoglobin gives you carbaminohemoglobin. This is totally incorrect, since we just mentioned that carbonic anhydrase actually converts carbon dioxide to carbonic acid. Therefore, we would only be looking at the products such as carbonic acid, hydrogen ions, or bicarbonate. Therefore, this is totally incorrect. The process of carbon dioxide combining with hemoglobin does not require carbonic anhydrase, therefore it won't be slowed down. 2. Carbon dioxide plus water forming carbonic acid. We just mentioned that this is correct, as you can see here, and carbonic acid dissociating into hydrogen ions. This is also correct, and the answer is going to be D. Question number 32. Which sequence of letters correctly identifies the order of events during the cardiac cycle? So let's start in a systematic way. We know that the sinoatrial node found in the right atrium initiates a wave of excitation. So the first step is going to be X. Now, once this happens, the atrial walls contract, because we just mentioned that the sinoatrial node is at the right atrium. Therefore, when the wave of excitation spreads, the atrium contracts. Then what happens after that is that this wave of excitation reaches the atrioventricular node. Now, the function of the atrioventricular node is to delay that wave of excitation a fraction of a second, so the atrium and the ventricles do not contract at the same time. Therefore, we have U. Once it entered the atrioventricular node, it got delayed. Now, what happens is once the wave of excitation exits the atrioventricular node, it passes through something called the bundle of His, found in the septum, and then once it reaches the bundle of His, it goes up again into the apex of the ventricle within the Purkinje or the Purkinje tissue. Now, once the impulse reaches the Purkinje tissue, the ventricles are able to contract. Now, the correct answer is going to be C. Question number 33. The diagram gives information about blood pressure in the left side of the heart during the cardiac cycle. Valves open and close at points numbered. Which row identifies the valves opening or closing at the points numbered? So first of all, there's a mnemonic that we must all know. It is CO, so a mnemonic once we see this graph of the cardiac cycle. So CO, CO, which indicates the opening and closing of the valves. Now, let's understand this further. So, the line at the bottom is the ventricle, and the line above is the aorta. Therefore, this is the semilunar valves. So here we're talking about the opening and closing of the semilunar valves, and at the bottom we're talking about the opening and closing of the atrioventricular valves. Now, let's see the suggestions that we have here. Which row identifies the valves opening? So, regarding the opening of the atrioventricular valve, it's at the bottom, therefore it's going to be 4. The semilunar valves opens at 2. The atrioventricular valves closes at 1. And the semilunar valves closes at 3. So, let's even understand this deeper. Now, regarding the semilunar valve, the reason why it opens is when it reaches the peak here, the pressure slightly increases. Now, once the pressure increases, it gives enough power or force in order to open the semilunar valves. And then once the pressure drops again, the semilunar valves closes, and vice versa regarding the atrioventricular valves. Therefore, the answer is going to be D. Question number 34. A person with no breathing conditions rests for an hour. Their breathing in this time is shallow and slow. Therefore, the volume of air is less. So little air from outside the body reaches the alveoli. The person's heart rate remains constant. Which statement is correct? Now let's see the suggestions that we have here. A. The carbon dioxide concentration in the blood in the pulmonary vein will be higher than in the pulmonary artery. So, carbon dioxide concentration is higher at the pulmonary artery because the pulmonary artery goes from the right ventricle into the lungs. And the reason why it does so is because it carries deoxygenated blood. And for it to carry deoxygenated blood, that means that the pulmonary artery contains a higher proportion of carbon dioxide since it contains deoxygenated blood. Therefore, the wording is totally incorrect. B. Carbon dioxide molecules in the air of the alveoli move out of the blood by active transport. So, carbon dioxide actually moves by passive transport. The reason for this is that carbon dioxide in the blood is within a higher concentration. Therefore, it moves out down concentration gradient into the alveoli, which contains a lower concentration of carbon dioxide. Therefore, this is totally incorrect. C. The air in the alveoli has a lower concentration of oxygen. This is incorrect. It would have a greater concentration of oxygen. D. Oxygen molecules diffuse from the air into the alveoli into the blood at a slower rate than when the person is active. This is correct, since when a person is active, a greater volume of air diffuses into the alveoli. Therefore, a greater concentration of oxygen diffuses. Therefore, this is correct, and the answer is going to be D. Question number 35, which statements about all bronchioles are correct? So this is mainly for memorization. You must memorize the components of each part of the lung, starting from the trachea and then into the alveoli. For one, they all have epithelium. So bronchioles do have columnar epithelium, so this is correct. Two, they have goblet cells. This is incorrect. They don't all have goblet cells. What has goblet cells is the trachea. Therefore, this is incorrect. Three, they have muscle tissue. Yes, they do have muscle tissue, and therefore the answer is going to be B. Question number 36. The diagram shows some of the pathogens that cause disease in humans and some of the ways they are transmitted. What is the correct pathogen and method of transmission of malaria? So, the pathogen that transports malaria is Plasmodium, which is a protoctist. Therefore, 2 is correct. Now, the method of transmission is via female Anopheles mosquito bite. Therefore, this is correct, and the answer is going to be 2x. Question number 37. Some of the processes which result in the formation of a population of bacteria that are resistant to a new antibiotic are listed. What is the correct order of these processes? First of all, there's a very common misconception that antibiotics cause resistance. This is obviously incorrect. What causes resistance is that there are normal mutations that happens throughout the process of the bacteria population formation. And then what happens is one of these mutations ends up being a resistance gene for a specific antibiotic. So once we have this clear, let's see the suggestions that we have here. 1. Change in reproductive success of bacteria, increase in frequency, increase in genetic variation, and random mutation. So as we said, we would always start with a random mutation. Occur that by accident contains a resistance gene for an antibiotic. Now, what happens is once there's a random mutation, mutations increase the genetic variation. So there's more genetic variation or more genetic material within the population, which we are able to choose from. Now, once this happens, what happens is the change in reproductive success of bacteria since the genetic material changed. And then finally, what happens is the frequency of resistance allele ends up increasing. Why does this happen? Because the bacteria which contains the resistance gene are able to actually survive whilst the others die. Therefore, the ones which are able to survive reproduce and increase in frequency, and this is where resistance happens. Therefore, the answer is going to be D. Question number 38. When bacteria are grown in a petri dish containing discs with antibiotics, there will be zones of inhibition of bacterial growth. Which conclusions can be made about the most and least effective antibiotics on this species of bacteria? So, first of all, we must identify how to interpret a petri dish. Now, first of all, here we can see an antibiotic disc. And right here is the area of bacterial growth. So it makes sense that the larger the zone of inhibition, the more effective the antibiotic. So, the larger the zone of inhibition, the more effective the antibiotic. Because if bacteria are able to grow right over the antibiotic, this means that the antibiotic is the least effective. So the less the zone of inhibition, the least effective the antibiotic. So let's see the suggestions that we have here. Therefore, the largest the zone of inhibition is going to be antibiotic 3. And the least effective is going to be the least zone of inhibition, which is 2. Therefore, the answer is going to be A. Question number 39. Which blood cell types does not recognize, engulf, and digest non-self particles? So, here we're talking about white blood cells which are not phagocytes. Now, phagocytes are macrophages, are monocytes, and neutrophils. So, macrophages are phagocytes, so these are incorrect. They engulf bacteria. Same with neutrophils. And phagocytes is a term composing of both, so this is incorrect. And T killer cells. So, T killer cells does not recognize, engulf, or digest since it's not a phagocyte. Therefore, the answer is going to be D. Number 40. Repeated infections with malaria result in more effective immunity to malaria. Which type of immunity is responsible for the more effective immunity? Therefore, here we're talking about a natural immunity since it got a repeated infection, so we're talking about the natural immunity. Now, with natural immunity, we must talk about the active immunity since there has been an infection, an active infection with malaria. Therefore, it's supposed to be a natural active. So the reason why the others are wrong, first of all, artificial immunity indicates that this is a vaccine. However, this is incorrect. So an artificial active are antigens found in vaccines which induce active immunity. So this is incorrect. And when we talk about passive, we're talking about antibody transfer, such as antibodies in breast milk. Therefore, this is incorrect, and the answer is going to be C. Thank you very much for watching this video. If you enjoyed this video, please give it a thumbs up and subscribe to this channel.